All right, welcome back, everyone. Today we're going to be looking at bone. We'll look at bone histology, or the osteon. We will look at three different types of muscles: smooth muscle, skeletal muscle, and cardiac muscle on the models pretty quick. We'll look at the skin model, the different layers of the epidermis and then dermis, and then all the different accessory structures that you can find deep in the skin. And then we'll look at the blood cell, and different types of white blood cells and the function of red. And that'll be it. Okay, so let's take a look. Let me do the screen share here for you. Okay, and let's look at, all right, so we've done the cell model, right? We did the microscope, we did the organelle, the cell, we did mitosis. So we're going to do bone histology models, the three types of muscle, the skin model, and then the blood cell. And then a separate video, we'll do the histology. Okay, we'll do a separate one just on that. So let's take a look first at bone. So when we look at bone, when you take any long bone, let's say we take the humerus, or we take the radius or ulna, even any of the phalanges, and we look at the compact portion, right? In bone, at the proximal and distal ends of any long bone, it's called the epiphysis. The proximal and distal end, where you have the epiphysis, is spongy bone. As you move towards the middle of any long bone, where the shaft is where it's compact. When I say compact, it sounds like it's solid, but it's really a tube. So it's kind of like it's got a wall type of structure all the way around. And that's what gives bones their strength, is that tube-like structure. If it was solid all the way through, you can actually break it easier. The fact that it's hollowed out with bone marrow in the center, but the outside portion is fully compact, it gives it a very unique structure, which makes it strong, but we call that an osteon. Osteons are only located where you find compact bone, and the visual appearance of an osteon would look like if someone cut a tree down, if they cut a tree in half and timber, right, it falls down, and you look at the stump, how it has all those concentric rings. You could say how old the tree is depending on how many rings it has. So osteons have a similar structure to that. All right, so the outer part of the bone that you can see is called the periosteum. And as you move deep inward, it's the endosteum. The periosteum is where muscles and tendons actually attach to bone. So muscles are the contractile part. When we say contraction, we mean tension can be generated in that part of the muscle. Muscles attach to bones by way of tendons. And tendons are kind of tensile. And when the contractile part of the muscle pulls, it pulls on the tendon, and the tendon moves the bone, right? So here on the right-hand side, in that aqua color, right, it says periosteum. That's the outer part of the bone. And as we move this way, we're moving to the deeper part. So this side would be the endosteum, all the way on the left, okay? You'll see that bone is vascular. There is this nutrient artery that penetrates all the way through, and then has these branches that move longitudinally, right, in the same plane as the bone. This one is kind of perpendicular to it, but all these run in the center, and you could see it in the center of each of these is what they call a Haversian canal in the center, where that artery would penetrate through. So this artery is going to bring oxygen, it's going to bring blood, it's going to bring calcium right into the bone. So you'll hear of these structures. We'll get a better look at these circumferential lamellae. It's probably not the best view to see them, but you could see that they're pulled upward, right? There are these concentric rings. And if we bring one of them up, So there's 3 of them here. They're called circumferential lamellae. I'll show you osteocytes and we'll explain what they are. But each of these rings going around is called a concentric lamellae. Concentric lamellae. All right, better view. It's a bird's eye view. So we're looking at this model, but now bird's eye view, looking flat down on it, straight down. So here, here, and here, those that are sticking upward are the circumferential lamellae. They're the outer ones, circumferential. All the way on the outside is the periosteum. It's kind of like grayish blue. That's the periosteum. Each of these is an osteon. So not each circle is an osteon, but if I take this that looks like a wedding cake, this entire structure is an osteon. Everything from here to here, this is all an osteon. This is an osteon. We have all these separate osteons, but they're made up of all of these concentric lamellae. These lamellae are all of those concentric rings, right? So here in the orange at top at around 12 o'clock, you could see concentric lamellae. If it completed itself all the way around, right, that would be another osteon. In the center of each osteon, so here and here is a central canal, also seen in purple here and here. That's the Haversian canal or central canal. There's going to be arteries there, veins, lymphatics, all running through the center. The outer part to the right, periosteum, all the way on the left, endosteum. Again, periosteum, you could see it, the outer part of the bone here. You could see the nutrient artery perforating and penetrating through. Osteocyte. So with bone cells, you have like a progenitor bone cell, which is a stem cell, which can differentiate into an osteoblast. And osteoblasts, with a B, build up bone. They're laying down matrix, calcium salts, collagen fibers. It makes the cement, and it's eventually the osteoblast, that immature cell, produces this matrix. So if I am an osteoblast and I'm an immature cell, but I'm producing the matrix of bone all around me, eventually that cement that I'm creating that's all around me is going to harden up. When it hardens up, I am trapped in my matrix, okay? I become trapped in my matrix, but I'm in now like this hollow cocoon, like in this cocoon, and it's a hollowed out cavity that I'm stuck in, but I can't get out. Now I'm trapped. The cavity that I'm trapped in is called a lacunae, and I'll show you what that is. I'm trapped in the space called a lacunae, but I can't produce any more matrix. Now I become a mature cell called an osteocyte. Okay, so right here, these black structures are osteocytes. Guess where they're trapped in? That little cavity called a lacunae. And the lacunae, in the lacunae, the osteocyte, me, I have now all of these finger-like extensions that are going to come out and create this matrix of communication with other osteocytes. Those are called canaliculi. Let's see if we could take a look at some of those coming up. Okay, so these are osteons, and in the center, you could see the Haversian or central canal. Each of these are concentric lamellae. When it's a full ring that makes an osteon, it's concentric lamellae. Otherwise, interstitial lamellae are between the full osteons. They're remnants of other osteons. So bone, like all other tissues in the body, they're made and broken down. They build up and break down. They build up and break down. Skin cells are made and then they break down and slough off. Hair is made and then they slough off. Nails are made and then you cut them off. Red blood cells are made and then in 120 days they die off. So bone goes through what's called remodeling phases, where it's made and broken down. The osteoblasts build up the bone, The osteoclast with a C breaks it down. So osteoblast builds up bone, osteoclast breaks it down. The remnant of some of that broken down bone is called interstitial lamellae. They don't form complete concentric rings anymore to make an osteon, so they're remnants. If you look in the concentric rings, you can see the lacunae. They're empty here. But guess what sits in those lacunae? This, an osteocyte. What was an osteocyte? What was this mature cell? At one point, it was immature. It was the osteoblast. The osteoblast makes the matrix. It gets trapped in the matrix. Now I become an osteocyte, and I'm trapped in this lacunae right there. Now I can take another look. We could see all these concentric rings. Number 4, they're called concentric lamellae. Number 3 is the Haversian system. In between the osteons are number 2, interstitial lamellae. They don't form complete rings. And then here, and here, and here, these are lacunae. The osteocyte is number 1. They have these extensions coming off of them that are called canaliculi. And we'll show them there are these things coming off. Those are called canaliculi. Again, you could see the Haversian canal or the central canal. You could see the osteocytes. They're stuck in their lacunae, and their hair-like extensions coming off of them are canaliculi. This is another bone model. It's yellow. It's not this one, but you could see same thing, concentric lamellae, these hollowed out canals. Those are the lacunae. What sits inside the lacunae is an osteocyte. See the blue nucleus surrounded by the red cell body. So that's the nucleus of an osteocyte. The canaliculi are these red spider-like, they make it look like a jellyfish that come off, but they're for the network of communication. Okay, there's the lacunae down on the bottom again. Again, you got your concentric rings, Haversian canal. You've got artery, vein, and lymph that are in the center of that. Here's a vein, here's an artery, here's nerve, all in that Haversian canal or central canal. Here's the lacunae in green. These are the concentric lamellae. Up above it, in between, interstitial lamellae. And here's the lymphatic vessel. You got lymph, nerve, artery, and vein. Hollowed out lacunae, osteocyte in the middle. The nucleus of the osteocyte is blue. The red extensions are the canaliculi. The cell body of the osteocyte and the nucleus of the osteocyte, and then the canaliculi. Okay, all right, so let's close out. That's the osteon. That's found only in compact bone. At the proximal and distal epiphysis, you do have spongy bone. Let's close that out. Let's look at the 3 different types of muscle. We have cardiac muscle, skeletal muscle, and smooth muscle. Let's look at smooth muscle, right? Where do we find smooth muscle? Look at the characteristics here. It's got some mitochondria. We don't see really any striations. The striations come from actin and myosin filaments. These are the contractile proteins. When light penetrates through actin and myosin through a microscope, it gives it a very striated appearance. So cardiac muscle is striated. Skeletal muscle is striated, but there are no striations in smooth muscle, which is why it's called smooth. It gives it a smooth appearance if you don't see striations. Where do we find smooth muscle? We find it around parts of the body that contract without voluntary conscious control. Arteries have to constrict and dilate. When you have an artery that vasoconstricts, blood pressure goes up. When it vasodilates, blood pressure goes down. Your intestines constrict and dilate. Your colon constricts, constipation, or dilates, too much, diarrhea. The bronchi, from your trachea, in the lungs, if you have bronchoconstriction, difficult to breathe, like asthma. Dilates, easy to breathe. So smooth muscle is around the trachea, the bronchioles, arteries, veins, all the organs, the uterus, anything that can contract, like the uterus, uterine contractions, right? That's smooth muscle. Skeletal muscle, you can see it's very striated because of all the actin and myosin filaments. Skeletal muscle is an all voluntary controlled muscles. You can voluntarily contract your biceps, your triceps. You can move, you can flex, you can extend, you can move your neck. All of these muscles are voluntary. They're all skeletal muscles, voluntary contractile, and it attaches to the skeleton. Skeletal muscle attaches to the skeleton. Then we have this muscle, cardiac, only found in the heart. That's why it's cardiac. It's branched, right? You can see that it's highly branched, like a yield sign. It is striated, but it has these intercalated discs. If this is a cardiomyocyte and this is a cardiomyocyte, like two combs, the teeth of a comb come together and they fit perfectly together. Where they fit, it's called an intercalated disc. That's where these blue lines are, purple. It's where the cells, the heart cardiac cells, come together. If we were to separate them and look inward in between, they're called gap junctions. When cells come together, they make tight junctions, they make gap junctions, desmosomes, hemidesmosomes, adherence junctions, all different type of junctions because cells come together to make tissues. So there's different type of unions that are created when cells come together. So intercalated discs are found only in cardiac muscle. You can see these black lines like this. Cardiac muscle also, lots of mitochondria because the heart is always beating. Anything that's always working has lots of mitochondria because it needs lots of energy. The brain, lots of mitochondria. Skeleton, lots of mitochondria. The liver is always working to detoxify, lots of mitochondria. Okay? All right, so those are the three types of muscle. Again, cardiac muscle is striated, but it's involuntary, right? You can't say, heart rate, go from 72 beats a minute to 90 beats a minute, like that. You can't do it. But you can have stressful thoughts, and over time, it can increase your heart rate. And then you can meditate, and over time, decrease your heart rate. But it can't be as quick as finger move, abducted shoulder, right? That's conscious, that's skeletal muscle. So skeletal muscle, cardiac muscle are both striated. Cardiac muscle is involuntary, skeletal muscle is voluntary. Smooth muscle, no striations, involuntary. Okay, let's take a look at skin. So when we look at skin, we have section, let's see if we could do this. If we look at Roman numeral 1, 2, and 3, we can see on the left-hand side, level 1, which is the pink and the blue, that's the upper portion, that is the epidermis. Number 1, epidermis. When we look at number 2, where it goes from the lower end of that purplish color all the way to the beginning of the fat cells, the adipose cells, that's number 2, that's dermis. That's where the majority of all the accessory structures lie, like the sweat glands and the oil gland and some of the nerve endings and the hair follicle, all in the dermis. Below that is the hypodermis, number 3, or the sub-Q, or subcutaneous. So up here, epidermis, down here, dermis, and where the yellow is all the fat, all the adipocytes, number 13, the adipose tissue, that's all hypodermis or subcutaneous. All right. The epidermis has several layers to it. They're typically called stratum layers. You see stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, and then layer 6 down here, that 6th layer is the stratum basal, or the basal layer. That's number 6. And then looks like 7 is the stratum germinativum, which would be all the way down on the bottom. So 6 and 7 are somehow intertwined together. The stratum germinativum, if you look at this, there's a little mnemonic here that says Cher loves getting skin Botox, which is the basal layer, sometimes called a stratum germinativum. So 6 and 7 are going to be interlinked, and it's that basement membrane portion, that dark area down at the bottom. All right. Cher, that's the C, loves lucidum. Cher loves getting granulosum. Spinosum. Cher loves getting skin Botox for the basal layer, stratum basal or the germinativum. The top layer is the corneum, number 2, the stratum corneum. That's like 25 to 30 layers of stratified squamous epithelium. We'll get into that in a little bit. That slough off, it's dead cells. All the way down here at the basement layer, at the germinative layer, you've got stem cells there. They're constantly dividing and multiplying. And as they divide and multiply, they push up, 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 and eventually become the stratum corneum. It takes about 4 weeks to get there, and then the stratum corneum sloughs off. And then the stratum basal layer, that germinativum layer, where all the stem cells are, they make more skin cells. Then they move up layer by layer by layer, and then they eventually slough off to become the stratum corneum. Okay, you'll learn a little bit more about the functions of these layers in lecture. Lucid means clear. The granulosum, there's granules in there that produce lipids and cholesterol and fat that keep the skin oily and moist. Spinosa means spiny. It's a spiny layer under a microscope. So Cher loves getting skin Botox are all part of the epidermis, the upper layer. When you get into the dermis down here, now we've got a few accessory structures. So let's take a look at that. Where is number 8? Number 8 is the papillary layer, which is up here. That's the upper portion. And then the reticular layer is number 10. That's the deeper portion. So you don't have many layers in the dermis. It's just papillary, upper, and then deeper reticular. Let's look at some of the additional important structures here. On the left-hand side, like here and here, the white and this white portion here is the sweat gland. You could see it here, you could see it here, and here it's cut through. The purple one, this is your oil gland, your sebaceous gland. We could see it here, 3D coming out at you, and we could see it here where it's cut. All right, and this is the hair shaft. And this is the erector pili. The erector pili muscle contracts, it pulls on the hair and makes the hair stand on end, which gives you like the goosebumps. It's just part of the sympathetic fight or flight response. So number 11 is your sweat gland that is known as the sudiferous gland. Sudiferous, S-U-D-I-F-E-R-O-U-S. Sudiferous is your sweat gland. Number 9 is the sudiferous duct. You can see it here, sudiferous duct. That's your sweat duct. Now over here, number 22, that's your sebaceous gland. Sebaceous, that's your oil gland. That's here and that's here. Then you have the erector pili muscle, which is here. Number 20. That's the erector pili muscle. We have some neurological structures here. Number 12, let me go to number Roman numeral 3 here, which is the subcutaneous or hypodermis. That's where you have this Pacinian corpuscle, number 12. Pacinian or Pacinian corpuscle, it's a deep pressure receptor. This picks up the sense of deep pressure, like when you're walking and your calcaneus hits the ground, or someone's poking you really hard with that deep, deep pressure. That's the Pacinian corpuscles. What else do we need to show you here? Here, these are the Meissner's corpuscles up higher. And then there's free nerve endings, which are like pain receptors. So number 1, right here, These are Meissner's corpuscles. These are more for like light touch, you know, very, very light touch, not deep pressure. That's Pacinian. Then you have free nerve endings here. Those are like your, for pain receptors. Okay, same type of structure here. Again, just a different plaque, 1, 2, and 3, epidermis, dermis, and subcutaneous. So on the right, number 1 is the epidermis. Here, this region is the dermis. And then you have the subcu or subcutaneous or hypodermis. In that hypodermis is where you have that Pacinian corpuscle. It's deep, your deep pressure receptor. As we go to the surface up higher, number 5, that's your Meissner's corpuscle. And then you have your free nerve endings. The yellow is adipose, which is fat cells. If we look upward here in the epidermis, this one's not showing all the different layers, or is it? Let's see. Oh, maybe it's showing it here, 25, 24, and 23. So 25 is the stratum corneum, stratum lucidum, and then the deeper stratum granulosum, and the deepest of all is 22, the germinativum. So if we look at 22, right, they're joining that one with probably number 20, which is the basal layer. That would be like 6 and 7 in the other slide. So right here is that basement, that basal layer that has the stem cells that move upward and eventually become the stratum corneum, and that sloughs off. That's made up of many, many layers of squamous cell epithelium, stratified squamous cell epithelium, because it's so many layers. Sebaceous gland, where is that? That is here, and it's also here, number 1. Let's look at the root of the hair, number 2. That's the root of the hair. 3 higher up, that's the shaft. 4 is the erector pili muscle. 5 over here is the Meissner's corpuscle for light touch. 6 is your sudiferous pore, the sweat. When the sweat gland opens up, that's the pore. 7 down here at the bottom, that's the Pacinian corpuscle. Don't worry about 8, 9, or 10, or even 11. You don't have to worry about the outer, any of those here, or the medulla of the hair and the hair cortex. Not really important. 14 you should know, that's the adipose or the fat cell. Number 16 you should know, that is the sweat gland, so that's here and that's here. 17 is the reticular layer, right there, of the dermis. And the papillary layer should be higher up, so that's over here, number 19. So you got the papillary layer, which is probably like the top 20%, and then the papillary layer, which is probably like the deeper 80%. Okay, so that's it. That's the skin. We'll close that out. And let's see if we could bring in the blood here. Let's look at the blood. Let's open this up, go over some of the keys here. So the primary function of blood is going to be to carry oxygen and CO2, right? So hemoglobin is going to bind to oxygen and carbon dioxide, and even nitric oxide. Nitric oxide is a gas that tells the blood vessel to dilate, okay? It's going to distribute nutrients throughout the body. It's going to be able to transport and carry hormones, and it's going to help to remove waste products from the body. The other function of blood is in our self-defense, in our immunity, because of the WBCs, or the white blood cells. It's going to help defend us against pathogens or toxins. Let's look at what blood is made up of. Blood is tissue. It's connective tissue. It's just liquid connective tissue. Tissue, when you look at connective tissue, it's got some cells with lots of matrix. The matrix is plasma. That's the extracellular matrix. The cells are the red blood cells, the white blood cells. So they are scattered throughout. Remember, epithelium was lots and lots and lots and lots of cells, with very, very little extracellular matrix. It's the opposite. Connective tissue is lots of matrix, little bit of cells. So the three types of formed cells that we have are erythrocytes, those are the red blood cells. We have white blood cells, those are leukocytes, the WBCs. And then we have, this should be thrombocytes with a TH, because thrombocytes create a thrombosis or a blood clot, and we call them platelets. So they used to be called thrombocytes, now they're called platelets. They're involved with clotting. If you take aspirin or baby aspirin, it blocks the platelets' function, and you don't clot as easily, and blood becomes very, very thin. Garlic can do the same thing. Niacin can do the same thing. Fish oils in high concentrations above 4,000 milligrams can also thin the blood. Garlic can thin the blood. Resveratrol or red wine can thin the blood. So all have a benefit. Okay. Let's take a look here. So when there's a blood draw, when someone draws blood, and where is blood drawn from? Is it drawn from an artery or is it pulled from a vein? Think about it. Where's blood pulled from? Arteries or veins? It's pulled from veins. That's why it's called venipuncture, veins. So we can extract from the plasma, which is about 55% of the blood. The cells are 45% of the blood. So what is connective tissue? Lots of matrix, 55%, with some cells, 45%. Within the plasma, you got proteins, you got solutes, and you've got water. Water makes up 92%. That's why drinking water is super important. How much water should we be drinking? You take your weight, cut it in half, drink it in ounces. That's the rule of thumb. Take your body weight, cut that number in half, drink that in ounces, and that is a sufficient amount of water that you should be drinking throughout the day. The formed elements are platelets, white blood cells, and red blood cells. Males have about 5 to 6 liters of blood roaming through their body at any given time. Women have about 4 to 5 liters of blood through their body at any given time. Okay, we're moving through this because I just want to get into the structure of them. What's interesting about red blood cells is that it's really not the best term to call it a cell, because cells have nucleus. A red blood cell does not have a nucleus. It actually loses it. So it should be like a red blood corpuscle. The reason why red blood cells don't have a nucleus is the largest organelle is a nucleus. We don't want red blood cells to carry a nucleus because it's got to carry hemoglobin. It's got to carry oxygen. It's got a lot of work to do. So it loses its nucleus in its developmental stages, okay? Red blood cells do not have mitochondria, all right? So all of their oxygen is obtained through anaerobic means. All of the oxygen that it delivers, that it is holding, it delivers. Okay, so you can see there's the venipuncture, the blood is pulled, it's centrifuged, the heavy stuff sinks to the bottom, the light stuff rises to the top. Plasma is the 55%, formed elements are at the bottom. So let's look at the formed elements. Here's the unique structure to a red blood cell. It's called biconcave disc because it's concave on the top. It's concave on the bottom. It's very flexible. The periphery is thicker. The center is thinner. So under a microscope, the center is hypochromic, less color in the middle, darker around the periphery. And if you look at the diameter, it's 7 to 8 microns. That's an important measurement, important number, because as red blood cells get older, between 100 to 120 days of age, they become less flexible. Much like a person of 120 years old, they probably lose their flexibility, right? So the red blood cells are going to move into a vessel. But this large diameter structure has trouble getting through a vessel half its size. So when it squeezes through there, it bursts. And that's how it gets recycled. Okay, so here's an RBC. All of these on the left, where it has the hypochromic center, these are all red blood cells. That's an RBC, a red blood cell. Now we have white blood cells. White blood cells can be categorized as granular or agranular. Granular, because if you look at the cytoplasm here, there's granules. The granular sites are BEN, B-E-N, basophil, eosinophil, neutrophil. Again, B-E-N, basophil, eosinophil, and neutrophil. We're looking at the neutrophil now. Notice all of them end in phil. From Greek, philos means friend of. Neutrophil takes a neutral stain. Basophil takes a basic stain. Eosinophil takes an acidic stain. So the cytoplasm granules all look a little bit of different color depending on the stain they take up. So what's interesting about a neutrophil is that the nucleus here, here, and here can have 3 to 5 lobes to a nucleus. That's a key feature. So here's 1, 2, 3 lobes. Here's 1, 2, 3, 4, 5 lobes of nucleus, very granular. Neutrophil fits that characteristic. The neutrophil is like the first line of defense. It's the first cell that arrives whenever there's an infection. When neutrophil count is high, there's typically a bacterial infection. When neutrophils are high, there's a bacterial infection. Here's the eosinophil. Look at the granules again. They're a little bit larger. They stain red, but look at the nucleus. It's only bilobed. When you see a bilobe nucleus, 2 lobes, it's an eosinophil. When eosinophil count is high, there's typically an allergy or a parasitic infection, usually allergies or parasites. That's when the eosinophil count is high. The granules are larger, they stain very red. And the reason for the granules is whether it's a neutrophil, eosinophil, or basophil, the granules degranulate. They're going to release some sort of enzymes or toxic stuff to kind of destroy the pathogen, okay? The basophil is a basic stain. It's dark, it's blue. Usually on the microscope, it's really hard to see the nucleus. It's somewhat obscured by all the dark staining granules. Basophils are going to be involved in releasing some histamine, and which is involved with inflammation. Basophils are also involved in allergies because basophils are releasing histamine, whereas the eosinophils that respond to allergies are releasing histamine ACE. Anything that ends in ACE is an enzyme. So the histamine ACE that the eosinophil produces goes after the histamine that the basophil produces to try and quench it, so you're not having such a crazy allergic reaction with sinus congestion and puffy and leaky eyes, right? So the granular white blood cells are the basophil, eosinophil, and neutrophil. Those are the 3 granular sites. The agranular means without granules, those are lymphocytes and monocytes. Granular sites, basophil, eosinophil, neutrophil. Basophil, the nucleus looks like an S. It's obscured by the dark staining granules. That's the basophil. The eosinophil, large red stained granules with bilobed nucleus. The neutrophil, the small red stained granules, and the nucleus has 3 to 5 lobes. Monocytes and lymphocytes. Notice no granules in the cytoplasm. The monocyte is kidney bean shaped. It's very large. Monocytes are always in circulation in the blood. When they find a problem in the tissue, the monocyte evolves into a macrophage. It's like a large Pac-Man. It's an eating cell. Monocytes become macrophages, which are large eating cells. Monocyte is a little bit smaller. You can see the nucleus. What's unique about it is the nucleus is almost as large as the cell itself. So here's the monocyte, very large, kidney bean shaped nucleus. Here's the lymphocyte, a little bit smaller, but the nucleus is just as large as the cell. When lymphocyte count is high, there's a viral infection. So with lymphocytes are high, it's viral. When neutrophils are high, it's bacterial. Just remember, never bet Las Vegas, NBLV. Never bet. Neutrophil is bacterial. Las Vegas. Lymphocytes high, viral. Never bet Las Vegas. Okay. And here's your platelet. Platelets are involved in blood clots. Okay. If someone's taking a blood thinner or aspirins to thin the blood to avoid clotting, it's platelets that do that. Aspirin blocks the mechanism. Okay. Here on this blood plaque, we can see a few cells. Let's look at number 1. These here, these are RBCs, red blood cells, also known as an erythrocyte. Where's number 2? Right here, 2 and 2, and the nucleus is just as large as the cell itself. Those are lymphocytes. When we look at number 3, it's agranular. This is a kidney bean-shaped cell. Number 3 is your monocyte. Number 4 is your neutrophil. 1, 2, 3, 4 heads to the nuclei. Let's look at this number 4. 1, 2, 3, 4, 5, and the one right here, 1, 2, 3. So those all have to be neutrophils. When they're a high amount, bacterial infection. Number 5, eosinophil. Here's number 5, 2 lobes. Now look, I would agree that this number 4 here and this number 5 look the same. So let's put a number 5 here and a number 5 here, and then everyone will be happy, right? So number 5 is eosinophil. Now I would agree that this number 5 here looks like it has 3 lobes. 1, 2, 3. If that's the case, this is a neutrophil. So real classically speaking, you should know that if I'm going to ask you for a neutrophil, it's going to be obviously 3 to 5 lobes. Those are the characteristics. If I'm showing you 2 lobes here in the center or here off to the right, eosinophil. All right, number 6 is your basophil, those dark blue staining granules. When you see the blue granules, that's going to be basophil. Number 7, these are your platelets for clotting. And then everything that's kind of like yellowish green in between, all of this extracellular matrix, is plasm. On a slide, these are red blood cells. This is bilobe, that's an eosinophil. 1, 2, 3, 4 to 5 lobes, that's a neutrophil. This is very granular, really obscured, hard to see the nucleus, gotta go with basophil. Here's a large kidney bean shaped, no granules, so it's agranular. It's either a lymphocyte or monocyte. Remember, lymphocyte's nucleus, just as large as the cell. Monocyte is the kidney bean shaped nucleus. Neutrophil again, 1, 2, 3, 4, 4 to 5 heads to the nucleus is a neutrophil. Here's your lymphocyte. There's the nucleus, just as large as the cell there. These are red blood cells all around. So what would number 1 be? Take a look. Think about it. What's number 1? What would number 2 be? I'll give you a hint. 1 and 2 are agranular, and 3, 4, and 5 are granular. So number 1, if you said it is a monocyte, you're right. What about number 2? If you said it's a lymphocyte, congratulations. Let's look at number 3. How many nuclei? 1, 2, 3, 4, 5. Which one is 5 nuclei with granular? If you said neutrophil, kudos, congratulations. What about this one? Dark staining, very dark, dark granules. The nucleus is a little bit obscured. That's your basophil, which means that this one has to be, when it's all red in the cytoplasm, it's your eosinophil, typically bilobed. So this would be 1 lobe, this is the other lobe. So it's still bilobed, it's only connected by this neck. That should be your eosinophil. Let's see if we're right. Monocyte, lymphocyte, neutrophil, eosinophil, and basophil. Fantastic. Okay, let's stop the share here. And I think we have finished that up. We'll close that out and stop the share. And I think we pretty much covered everything. Let's make sure. There's still one more presentation left, and that will be on tissues. And I'll probably divide that up a little bit. But everyone's doing well so far. Keep up on your studying. It's really important to look at this material, you know, every day, a little bit every day. Just keep looking at it, get familiar with the language, look at the pictures over and over and over again, and you should do fine. Anatomy and physiology is really all about repetition, repetition, repetition. If you don't look at it enough, you're going to stress out. You can't just look at it the day before the test. You got to look at it over and over and over and over and over and over and over. All right? All right, everyone. We're going to bring this to a close. We hope you enjoyed that lecture, that lab.